Most people picture calories as a straightforward deposit. Eat a certain number, and that number sits in some kind of internal ledger, waiting to be spent on walking, thinking, or just staying alive. What almost nobody accounts for is that the deposit itself isn't free. Turning food into something the body can actually use requires machinery, and machinery runs on energy, which means a chunk of every meal gets spent before it ever becomes fuel for anything else. This isn't a footnote buried in some obscure corner of physiology. Depending on what's on the plate, this hidden toll can swing by a factor of 10 or more between one type of food and another. 2 meals with identical calorie counts can leave the body with meaningfully different amounts of usable energy left over, purely because of what those calories were made of. By the end of this video, all 3 macronutrients will be ranked from cheapest to most expensive to process, and the gap between the top and the bottom isn't small. The explanation isn't about willpower or metabolism in some vague sense. It comes down to a very specific piece of biochemical machinery tucked away in the liver, one that most people have never heard of. And it's the real reason this ranking looks the way it does. The phenomenon has a name, and it's older than most people would guess. In the 1880s, German physiologist Max Rubner noticed something strange while measuring heat production in animals after they ate. Metabolic rate jumped well above resting levels, and it stayed elevated for hours. He called it spezifische dynamische Wirkung, which loosely translates to specific dynamic action. Today, it usually goes by a more descriptive name, diet-induced thermogenesis, but the observation Rubner made more than a century ago still holds up. What he was picking up on is simple in concept. Eating raises the body's energy expenditure above what it would be during a fast, and that rise isn't caused by activity or digestion in some vague sense. It's caused by real, measurable biochemical work. Textbooks tend to summarize the overall effect as a modest slice of total daily energy expenditure, but that single figure hides enormous variation depending on what's actually being processed. That variation is the whole story, and averaging it away misses the point entirely. A number like roughly 10% describes a mixed plate of food, not any one nutrient on its own. To understand why some meals cost so much more to process than others, the conversation has to leave the dinner table and head somewhere far less visible. The exact molecular steps involved in breaking a nutrient down and building something new out of it. Here's a fair question. How do scientists even measure a cost that never shows up on a scale or in a blood test? The answer is a method called indirect calorimetry, and it works by never touching heat directly at all. Instead, it tracks how much oxygen a person consumes and how much carbon dioxide they produce, breath by breath, and uses those two numbers to back-calculate exactly how much energy the body is spending. The ratio between those two gases, carbon dioxide produced divided by oxygen consumed, is called the respiratory quotient, and it acts like a fingerprint for whatever fuel is being burned at that moment. In 1983, physiologist Keith Frayne published a set of equations in the Journal of Applied Physiology that let researchers convert those raw gas measurements into precise oxidation rates for fat and carbohydrate. Those equations are still the standard tool used today. Decades later, whenever scientists need to know exactly what a body is burning and how fast. To isolate the cost of digesting a specific meal, researchers measure a person's energy expenditure while fasting, then measure it again after they eat, and track the difference over the following hours. The gap between those two lines on a graph is diet-induced thermogenesis, made visible through nothing more than exhaled air. And once that measurement tool existed, scientists could finally start asking the more interesting question: what happens differently inside the body depending on which macronutrient just came through the door? Here's where the story takes its first real turn, and it's not the one most people expect. Breaking a protein apart into individual amino acids technically called hydrolyzing its peptide bonds, is not actually expensive. Enzymes like pepsin and trypsin cut those bonds through a downhill chemical reaction, one that doesn't require the cell to spend a single molecule of ATP to make it happen. The enzyme just speeds up something that was already thermodynamically favorable. So where does protein's reputation for being metabolically costly actually come from? Part of it lives in the stomach itself before a single amino acid is even absorbed. Producing the hydrochloric acid needed to unfold proteins and activate digestive enzymes requires a dedicated pump embedded in the lining of the stomach, the H+, K+ ATPase, which spends ATP to shove hydrogen ions into the stomach against a gradient over 1,000,000 times steeper than what exists inside the cell. That pump has to run constantly to keep the stomach as acidic as it needs to be. Then there's the trip across the intestinal wall. Amino acids don't just drift into the bloodstream. Most of them are hauled in through sodium-coupled transporters, a process called secondary active transport. That system only works because a separate pump, the sodium-potassium ATPase, is continuously burning ATP in the background to maintain the sodium gradient those transporters depend on. Breaking protein apart may be cheap, but everything surrounding that step, acid production and absorption, already carries a real energy price tag before amino acids have gone anywhere useful. This is where protein's real cost shows up, and it has almost nothing to do with breaking anything down. Once amino acids reach the bloodstream, the body doesn't simply file them away for later. A large share gets used immediately to build brand new proteins, a process called protein turnover. And building something from scratch is a fundamentally different kind of expense than taking something apart. Assembling one peptide bond between two amino acids costs 4 ATP equivalents at the ribosome. 2 spent charging the amino acid onto its transfer RNA at an enzyme called aminoacyl tRNA synthetase, and 2 more spent on GTP hydrolysis during the elongation steps carried out by factors known as EF2 and EFG. In a landmark 1995 paper published in the Biochemical Journal, researchers Frank Butgereit and Martin Brand ranked the major energy-consuming processes inside a living cell, and protein synthesis came out on top of that hierarchy, ahead of nearly everything else the cell spends energy on. Compare that to what's coming later in this video for carbohydrate and fat, and the contrast is stark. Storing glucose means forming a single glycosidic bond. Storing fat means attaching a fatty acid to a glycerol backbone through esterification. Neither one involves anything close to the 4 ATP per unit price tag that comes with rebuilding a protein 1 amino acid at a time. That difference in raw construction cost is the first real clue toward why protein sits where it does on the final ranking. Not every amino acid that gets absorbed ends up as part of a new protein. Many are broken down for fuel or converted into other molecules, and that process strips off their nitrogen-containing amino group, leaving behind ammonia, a compound that's genuinely toxic if it accumulates. The body can't simply exhale it the way it exhales carbon dioxide from burning carbohydrate or fat, so it needs a dedicated chemical assembly line to convert that ammonia into something safe to excrete. That assembly line is called the urea cycle, and it was worked out in 1932 by biochemists Hans Krebs and Kurt Henseleit, using nothing more than slices of liver tissue and careful measurement. It holds a special place in biochemistry as the first metabolic cycle ever mapped out in full, years before Krebs went on to describe the citric acid cycle that carries his name today. What he and Henseleit revealed was a 5-step loop running mostly inside liver cells that takes 2 nitrogen atoms and packages them into a single molecule of urea. That packaging isn't cheap. Producing 1 molecule of urea costs the equivalent of 4 high-energy phosphate bonds, drawn from 3 separate ATP molecules. Broken down further, that works out to roughly 2 ATP equivalents spent just to dispose of a single nitrogen-containing amino group. Neither carbohydrate nor fat forces the body into anything resembling this cleanup operation, because burning sugar or fat for energy leaves behind nothing more troublesome than water and carbon dioxide. Protein is the only one of the three that comes with a built-in waste disposal bill attached. If you've made it this far, drop a comment. Which of these hidden costs caught you off guard the most? The stomach acid, the protein rebuild, or that nitrogen bill only protein has to pay? Because the story is about to shift gears, and carbohydrate is about to show how it pulls off almost the same job for a fraction of the price. Physiologists split the heat generated after eating into two separate categories, and the distinction matters here. One is called facultative thermogenesis, the kind of heat production that can be dialed up or down depending on things like ambient temperature or the activity of brown fat, driven largely by the sympathetic nervous system. The other is obligatory thermogenesis, and this is the category that everything covered so far actually belongs to. Obligatory thermogenesis refers to the fixed, non-negotiable energy cost of digesting, absorbing, and storing whatever nutrients just came in. It doesn't fluctuate much with mood, temperature, or willpower because it's baked directly into the chemistry of the processes involved. The proton pump firing in the stomach, the sodium-driven transporters hauling amino acids across the gut wall, the ribosome spending 4 ATP per peptide bond during resynthesis, and the urea cycle disposing of nitrogen afterward. All of that machinery runs automatically every single time protein is consumed, whether the body wants it to or not. This is precisely why protein's cost is so hard to dodge. It isn't one expensive step tucked away somewhere. It's an entire relay of ATP-consuming reactions stacked one after another, each one obligatory rather than optional. Carbohydrate and fat trigger a much shorter version of that same relay, which is exactly what starts to become clear once their processing gets examined step by step. Glucose enters the bloodstream through a process that looks superficially similar to how amino acids get absorbed. A transporter called SGLT1, sitting on the surface of intestinal cells, pulls glucose in alongside sodium ions, riding the same sodium gradient that the NAK-ATPase pump works to maintain in the background. So far, the entry cost isn't wildly different from proteins. Where the paths diverge sharply is what happens after glucose reaches the liver or muscle and needs to be stored rather than burned immediately. Storing it means linking one glucose molecule to the next, forming what's called a glycosidic bond, the chemical connection that strings individual sugar units together into a long branching chain called glycogen. That single bond-forming step is the entire storage mechanism. Nothing close to the multi-stage relay protein goes through. There's no separate disposal system required afterward either. Glucose doesn't carry a nitrogen atom that needs to be stripped off and shipped out through a dedicated cycle. Whatever isn't stored as glycogen or burned right away for energy leaves behind carbon dioxide and water once oxidized, the same clean exit fat uses, and neither of those byproducts demands anything like the urea cycle's 4 ATP toll. Building glycogen isn't entirely free, though it's worth putting an actual number on how far below protein's cost it lands. Adding one glucose unit onto a growing glycogen chain costs 2 ATP equivalents, one to convert glucose 1-phosphate into its activated form, UDP glucose, using a molecule of UTP, and a second to regenerate that UTP afterward. The enzyme glycogen synthase then forms the actual bond, stringing the newly activated glucose onto the chain. 2 ATP per unit sounds substantial in isolation, but context changes everything. 1 gram of glycogen, once fully broken back down, yields roughly 30 ATP molecules through complete oxidation. Spending 2 of those 30 upfront on construction works out to something in the neighborhood of 5% of the stored energy, a noticeable dent, but nowhere close to the 4 ATP protein spends per single amino acid. On top of everything else protein's pathway requires before that step is even reached. That 5% figure also stays remarkably constant regardless of how much carbohydrate gets eaten in one sitting, because the chemistry doesn't change with quantity. There's no equivalent of the urea cycle waiting in the wings to add another charge on top. Glucose gets absorbed, gets linked into a chain, and that's essentially the whole transaction, a strikingly short story compared to the one protein tells. Fat's journey through digestion looks different from the very first step. Dietary triglycerides get emulsified by bile acids and broken apart by pancreatic lipase into monoglycerides and free fatty acids. And unlike amino acids or glucose, a large share of what gets absorbed simply diffuses across the intestinal lining rather than needing a dedicated ATP-hungry transporter to haul it in. There's no equivalent of the sodium-coupled pumps working overtime in the background. Once inside the intestinal cell, those fatty acids and monoglycerides get reassembled into new triglycerides for packaging into chylomicrons, and that reassembly does cost something. An enzyme called fatty acyl-CoA synthetase spends roughly 2 ATP equivalents to activate each individual fatty acid before it can be attached to a glycerol backbone through a process called esterification. On paper, that's the same rough price tag as adding 1 glucose unit to glycogen. The difference is what that 2 ATP charge is buying. A single fatty acid molecule is a long chain of carbons capable of yielding well over 100 ATP once it's eventually broken down for fuel, far more energy than 1 glucose unit provides. Spending 2 ATP to activate something that valuable barely dents the balance sheet, which is exactly why fat has earned its reputation as the cheapest of the 3 macronutrients to put into long-term storage. Lined up side by side, the pattern isn't subtle. Protein forces the body through acid secretion, active transport, ribosomal resynthesis at 4 ATP per bond, and, for whatever nitrogen isn't reused, a urea cycle charging another 4 ATP per molecule disposed. Carbohydrate involves a similar entry cost through sodium-coupled transport, followed by a comparatively modest 2 ATP to form each glycosidic bond with no disposal system required afterward. Fat skips most of the transport cost entirely and spends a small fixed activation fee on a molecule that returns an enormous amount of energy once it's actually used. None of this is a matter of opinion or dietary philosophy. It's arithmetic, written directly into the biochemistry every cell in the body runs constantly, whether anyone is paying attention or not. The reason protein carries the heaviest processing cost of the 3 was never really about protein being harder to digest in some vague sense. It's the compounding effect of several genuinely expensive, entirely obligatory reactions stacked one after another, with nitrogen disposal as the final, uniquely protein-specific charge tacked onto the bill. What time and mainstream nutrition are the same and where they diverge? Get down here. The hierarchy of cost sits protein highest, carbohydrate next, fat lowest. And that ranking exists for reasons that have nothing to do with calories in versus calories out, and everything to do with what's happening at the level of a ribosome, a transporter, and a 5-step cycle discovered almost a century ago in a lab slicing liver tissue by hand. If this kind of deep dive into what's actually happening inside the body earned a like, tap it, subscribe for more of this, and share it with someone who still thinks a calorie is just a calorie.